after the board trustee meeting or something like that. So I'll take you through this chapter 13. I have no idea how your regular lectures go, so you're just going to hear mine. And I'll give you my spin on things. In the back of the PowerPoints, I've got some recommended questions that I've sent to your professor so that she can see what I tell my students, at least in the book, they should be looking at. I hope you read the chapter. It's really a fascinating subject. Years ago, I did a master's in microbiology and virology, so it was something that was, I found fascinating early on in my career. Just for who I am, I teach microbes here. I've been teaching for about 5 years now. I retired from the Centers for Disease Control and worked at CDC for about half of my career. Other half of my career was with Johns Hopkins University and University of Maryland. Between those three places, that's pretty much where I spent my career. The last 7 years of my CDC career, I was stationed in Africa, in Europe, in a variety of places because I was assigned to the World Health Organization, which was really neat. So some of the stuff that your book talks about, for instance, in DRC, Democratic Republic of Congo, it talks about an Ebola outbreak. Well, I was in Zimbabwe for 3 years. I was a regional advisor, and indeed, we had a lot of problems. And we worked with the South African government because they had very good diagnostic techniques and laboratory infrastructure. We worked with DRC, Ebola outbreaks. We had Uganda, there was a Ebola outbreak that we worked on. So this is not far away, and it still happens. And what you're going to learn today, the overview of this viruses. I'm not really going to talk about viroids. Those are plant viruses, and none of us are plants, at least not today, and so we won't cover those. But let's get a little background here. If you print it off and you've got something to write on, that helps, I think. Viruses are different. They're very different. They cause many infections in humans and animals and plants and bacteria. You see, they can't carry out any metabolic pathways. They cannot. They don't grow and they don't respond to the environment. Maybe like somebody you know. They can't reproduce independently, and they are obligate intracellular parasites. Now, you think of parasites as, oh, you mean like a parasite, one of these things, an amoeba or a, well, that's, the word parasite is used. We can have bacterial parasites, we can have the protozoan parasites, the worm parasites. So when I say parasite, I don't mean parasite like you may be thinking. I'm talking about there's an organism that is living inside and must live inside a cell. One of the things that characterizes viral diseases is that they are still a plague of the industrialized world. Maybe a viral disease that certainly affects the United States. HIV. Another.
Flu.
Flu, influenza. Yeah. Another. What?
Human
Most
population.
STDs.
HPV.
Yeah, HPV.
Most
Human
STDs.
papillomavirus. Sure. Meningitis. Yeah. West Nile virus. You know. It's interesting because a lot of the bacterial organisms and pathogens that we deal with, we don't have a huge problem with. Yeah, we have problems periodically with foodborne outbreaks and things like that. On shipboard, you might have a bacterial infection, you might have it in food. But viruses, think about cruise ships. Cruise ships are notorious for having a diarrheal episode that basically shuts down the cruise ship. Norovirus. Is it because cruise ships are inherently dirty and that they are really scuzzy? No, I've been on a tour of a cruise ship food preparation area, and I'll tell you, they are wonderful. It's the fact that the norovirus is a very easily transmittable human virus that will be transmitted by fomites. Fomites is a term we use for anything that's inanimate that can transfer disease, such as a doorknob, a slide clicker, et cetera, et cetera. There's lots of fomites, and noroviruses are transmitted by fomites. And here's another thing with norovirus. It doesn't inactivate very rapidly. Now, I said inactivate. What would be the parallel word in other organisms to inactivation with viruses?
Wait, what?
Yeah, it's a hard question. I say inactivation of viruses because viruses don't die. They're not living. Viruses are not living. Okay? They're acellular, which means they don't have a cell. By the way, the things that I put in red, I tell my students, latch on to that. It's not the only important thing on the slide, but it's sort of key issues that you should kind of circle or, maybe perk up. Viruses are acellular. They do not have a cell like a bacterial cell. They're infectious agents that have either DNA or RNA, but never both. They could be RNA viruses, DNA viruses, but you never have RNA DNA. There's no cytoplasmic membrane, no cytosol. What's the cytosol?
The skeleton.
Cytosol is...
The skeleton.
That's that jelly-like stuff inside of cells. In a bacterial cell, you have that milieu of stuff that things are floating in. That's the cytosol. Well, they don't have that. They don't have a cytoplasmic membrane. They don't have organelles. And your textbook says there is an exception. Basically, they don't have organelles. They have both extracellular and intracellular states. Okay, that means they can be inside of a cell and exist, and they can be outside of a cell and exist. Well, we need to get the vocabulary of virology. And the vocabulary, like every vocabulary, is something you have to learn. It's different. The words may not sound terribly familiar, or they may, and they're confusing. A virion is a virus particle outside of the cell. Okay, a virion. So if we talk about a virus particle, we may say, here is influenza virion. What am I talking about? I'm talking about that whole structure. The capsid is the protein that surrounds this virus. Okay, surrounding basically the nucleic acid. That's all it is. It's nucleic acid with a protein coat. So the capsid is this protein coat. The nucleocapsid in the architectural viruses is the combined nucleic acid, DNA or RNA, and this protein. Okay? And then finally, the envelope is not all viruses have an envelope, but some viruses do. And it's a phospholipid layer that encloses this capsid, okay, this protein coat. And we're going to talk about this envelope because it's really an important part of the viruses that have it. Now, talking about viruses, the outermost layers provide protection and recognition. And I put down specific binding sites. You think of viruses and the kind of viruses there are. There's a hepatitis virus. What would it attack? What would it attack? Hepatitis virus. Liver. Good guess. No, you didn't. Hepatitis virus. How does a hepatitis virus know to go to the liver? Why doesn't it infect your lungs?
It has the envelope from the liver tissues.
They have what?
Envelope.
The envelope.
Their envelope has receptors for
they had They have on the outermost layer. They have specific receptor sites. Let me give you an example. A respiratory virus. Name a respiratory virus.
Tuberculosis.
Wrong. That's a bacteria.
That's a bacteria.
That's Mycobacterium tuberculosis. Yes, it's a respiratory organism. What is a very common, we get a shot for it every year, it is the flu virus.
Flu virus.
Flu virus. Ah, yeah. Flu virus, okay. So respiratory virus, where does that infect?
The lungs.
The lungs, the epithelium, the ciliated epithelium of the bronchial tree, your throat.
No.
This outermost layer with these specific receptors, I should put that in red, is very important because it helps, it doesn't help, it causes that virus to be specific for a specific kind of cell that it will infect. And that outermost layer provides the specificity for what a virus attacks. And we'll talk more about that. Inside of a cell, the capsid, this protein coat, is removed. What's left? Just the nucleic acid. That's the only thing that's inside a cell. When it's in, the only thing is the virus, and when part of the virus inside a cell is just the nucleic acid. That's all there is. So, now let's stop. Let's not go there yet. I want you to think of a virus as an organism that is able to hijack the cell that it infects. That's a good analogy. It comes in and it hijacks the cell, takes it over. Now we said it doesn't have any of the organelles, the virus doesn't have any of the organelles, doesn't have any of the structures, it doesn't have anything. All it is either RNA or DNA, and it codes for its own proteins, its own receptors, its own nucleic acid, to the detriment of the host cell. It comes in and it takes over. And it kills the host cell. Because what happens? The host cell, when it's infected, stops making the normal things that it would make, and it starts making viral particles. Here's a go. When I was in graduate school, they gave me an exam. There were 5 questions on this exam. It was at San Diego State University, where I was trying to get into the master's degree. And they asked us 5 questions. I had a bachelor's in microbiology, so I had a little bit of the background, which was fortunate. And in your written pre-qualification oral and written exams, you had to give them certain indication that you could go into the program. You had a little bit of a background. One of the questions they asked me was, is a virus the most advanced pathogen, or is it the most primitive? Do something think about that. Is it the most advanced, or is it the most primitive? You see, some people look at viruses as very, very primitive because they don't have all this other stuff that eukaryotic cells have, that even prokaryotic cells like bacteria have. All they have is a protein code and a nucleic acid. And some have said, well, they are very primitive. Others have said, no, it's just the opposite. Those are super advanced. And they have done away with the need of all this other stuff, and they hijack the cell, and so they are more advanced than the cells that they infect. Now, that's a rhetorical question in the sense that you could write either way and not be wrong. But whichever way you chose, you had to write, and you had, I don't know, they gave you reams of paper, they said you've got 5 hours. It was actually, it was 4 hours in the morning, 3 hours in the afternoon, so 7 hours, and we had 5 questions, and we just had to write what we could. So, is a virus really, really advanced, or is it really, really primitive? And that's a good question. Well, here's how we distinguish viruses. And we have to do this because they're exquisitely small. They don't have a lot of characteristics. You know, you can't say, well, he's 5 foot tall, blue-eyed, and brown hair, and, you know, whatever. So the first thing is RNA and DNA, and the number of strands. Are they single-stranded or double-stranded? RNA or DNA. The kinds of cells that they attack, and I mentioned this before, the term for that attractiveness, a hepatitis virus for a liver cell, a influenza virus for the ciliated epithelium of the respiratory tree, that's called a tropism. Tropism. It attracts, if you will, this virus to this particular tissue. The size of the virus, that's another way we can characterize it, we do. The characteristics of the protein coat, or the capsid, you'll see there's certain characteristics. The overall shape of the virus, and the presence or the absence of an envelope. Not all viruses have an envelope, but those do. We'll look at some of those. So these are some of the ways that we characterize and try and make distinctions. It's like gram-positive cells, gram-negative cells. Yeah, it's artificial, but it helps us to divide broad categories of things, and then in the gram-negatives we can go into the Enterobacteriaceae, the things that cause infection in our intestines. We can go into this, this, and this. And in the gram-positives we can go into the cocci, streptococci, staphylococci, and things like that. So it helps us. Okay. I mentioned the genetic material is either DNA or RNA. This is the primary way that we classify viruses. If you were to take virology, they would probably break the course into two halves, RNA viruses, DNA viruses. Then you would study the diseases and the characteristics, because it was a medically oriented course, of these two different types of viruses. And I said they could be double-stranded or single-stranded, either DNA or RNA. Rest of the stuff, yeah, the viral genome. Familiar with the term genome? What's the genome?
The entire genetic information of a single organism.
Exactly. Genome is everything. It's the genotype. We're talking about the genotype. It's everything genetic that makes up the organism. What's the phenotype? Genotype, phenotype.
Physical expression.
So,
Physical expression.
Physical expression, exactly. It's the physical expression of what we can see. Another chapter we talked about genetics, and you know about 25% of genes are not expressed. They're in our genotype, but they're not in our phenotype. You have genes that are switched off, unless you're still in your growth spurt, because you're not making growth hormone. Neither am I. My growth hormone stopped 100 years ago. There's genes that are never expressed. They're cryptic, if you will. The viral genome is much smaller, much, much smaller. And than the genome of bacterial cells and eukaryotic cells. I said that right. Okay. Specificity. They're specific, as I've mentioned, for a particular type of cell. What was the word? Started with a T?
Tropism.
Tropism. Tropism. Traction. You may have an tropism for somebody else, right? Well, that's what these guys are. It's a tropism. It's an attraction form. Most viruses have a specificity for a single common cell. And that affinity is due to receptors on the viral surface and the cell surface. There's a receptor there on one that matches the receptor on the other. So there's attachment proteins. These attachment proteins are also often called and often composed of glycoproteins on the cell surface. Viruses that infect many kinds of cells and many different hosts. So there are specific and there's general. Specific viruses for specific cells, general. Think of a virus that attacks an animal and also attacks humans. HIV, that's true,
Yeah.
because there are simian retroviruses that are very, very simple. In fact, they can be humanity as a model for human HIV infection. Okay, think of something else.
Rabies.
West Nile, yeah, West Nile, that's true. Rabies virus, yeah, there's a lot of viruses that animals can get, and we can get because we have the same receptors. Now, if you have a dog, like I have a dog, your dog has to be vaccinated against, say, canine distemper, but not, you don't get canine distemper. You don't get feline leukemia, but your cat does. And that's because your cat, your dog, has different receptors. But both of us, cats, dogs, and humans, we all have neural tissue, okay, neurons, and we can get rabies. And so if we have the same kind of cell that a virus in general, with a general ability to infect, yeah, then we are at risk. And you've named good examples of zoonotic diseases. Zoonotic is any kind of disease that goes in the animal. Well, Ivan Aspen, back in 1892, a few years ago, he discovered the first, what he called a virus. Now, when he used the word virus, he was actually using it as meaning a poison. He wasn't using what we know the word to mean today, this unique entity that can cause disease. Ivan Aspen found that you could take a tobacco plant, you could mince it up, you could filter it through the best filters that they had, and this was a sintered glass filter. It's sort of like, it's porous glass and has very, very small pore size. Takes all the bacteria out. In fact, everything known that was living and infected was taken out. And so they said this tobacco mosaic virus was non-filterable, which means it didn't get stopped by a filter. They thought it was a poison, and these days they were using the term virus to mean generally something that was poisonous. But actually what he found is this organism, this virus, tobacco mosaic virus, if you put it on the leaves of a healthy tobacco plant, it did this bad thing. And that was how they studied. Did they know what it was? No, they had no idea. Didn't have a clue. Didn't have a microscope or an electron microscope to be able to see it. So it was quite a few years later before somebody really said, oh, okay, this non-filterable poison or virus, if you will, is a unique entity, and it's different. Okay? This is a E. coli. This is a big thing. Okay? Big lumpy thing. These are bacterial viruses that are attached to that E. coli cell. Okay? These are called the T-even phages because there's T4, T2, T6, T8, et cetera, et cetera. They look to me like a little lunar lander. Okay? And this thing, this is a bacteria cell. Here is this bacteria phage. What does that mean?
Kill.
Say again?
To kill.
No.
Phage.
Bacteriophage, phage comes from the Latin word to eat.
Eat.
Dysphagia, the inability to eat. Phagocytosis, that comes from this root word means to eat. Literally, bacteriophage means a bacteria eater. Okay? A bacteria eater. And this thing, bacteriophage, now we would call a bacterial virus. Okay? So viruses that attack bacteria are called bacteriophage. They specifically attach to this. You can see the size differential here. I mean, it's marked. These you have seen in lab with 1,000 power in your microscopes. You've seen bacteria, and they look pretty small. they're just little blue or little pink, red dashes. They're pretty small. Well, if you could blow that tiny little bacterium up this size and see this, then you'd say, whoa. And there are viruses that are much, much smaller than this. The poliovirus, for instance, really super small. Let's look at some more. Viral hooks, animal cells. This is an influenza virus. And this is a cell, this is what I did my master's degree in, San Diego State. Using influenza viruses to infect chick embryo fibroblasts, because the chick embryo was a good model for us to grow these influenzas in. And what happens, and you'll see it more later, these viruses bud out of the cell that they are growing. They are enveloped viruses, and as they bud out, they take a piece of the envelope of this cell and they wrap it around themselves. Okay? So they do have an envelope. More about that later. That's influenza virus. Here's an HIV. HIV, this is a false color, by the way. It will actually look like this, but it's showing colors due to different proteins and different topography of the virus. This is a white cell. What does the white blood cell? The cells that these guys are specific for are, the T4s.
Yeah.
Okay? CD4, excuse me, CD4, I'm saying T4, CD4 white cells, they're white cells that basically are controlling the expression of the immune system. You kill those CD4 white cells, and you basically cause the individual to become
vulnerable. Yeah.
What do you die of when you die from AIDS?
Anything.
Secondary
That's
infection.
right. Secondary
They're vulnerable.
Anything can get you, and usually anything does get you if you have that. There's ways that we can look at these white cells and we can see when a person becomes immunosuppressed, because you can be infected with an immunodeficiency virus and not have AIDS. AIDS is a clinical classification. When your CD4 count drops below 200 per deciliter in your blood, you become immunosuppressed. You cannot fight off your other invaders, bacterial, virus, parasitic, you name it. So these guys here, they're looking specifically, and in my class, when we get into STDs and we talk about this, we talk about these receptor sites on the white cells. We talk about the binding on this and why this is such a difficult virus. You know, we have known since 1982 that it was a retrovirus, but we still don't have a vaccine. Why? Well, it's like malaria. We've been working on the vaccines for years, and we still don't have a good malarial vaccine. There's some bright spots on the horizon, but this is a very complex infection. Okay, so here's HIV attaching to white blood cells. Size of virus we mentioned. This is a red blood cell. This is a bacterium. You can see, you know, red blood cells, you see those, and they're pretty small. If we take that with the bacterium there and we make this bacterium that big, then we can start to see viruses. And then if we make that bigger, we can see that this virus here, it's a smallpox virus. We can see the bacteriophages. This is one of those T evophages of E. coli. That looks like a lunar lander, man. And indeed, it's got all these structures, a base plate, it's got a column, it's got a head, the genetic material is in here, and it basically injects the genetic material into the host cell. These little tail fibers have receptors on them. So that thing comes down and actually is looking for a receptor, say if this is an E. coli, that is very specific for this bacteriophage bacterial virus to land on. I said the poliovirus, and where is it? Yeah. Poliovirus is here, this one over here. That's another kind of bacteriophage, but poliovirus is a human virus. Look at the size of this thing, 30 nanometers. And that is really, really small. If you look here, look at nanometers. Now, this is 750. Well, this guy here is only 30. Only 30 nanometers. By the way, just an interesting aside. When I was with the World Health Organization, part of my job was bioterrorism preparedness. And how do we keep, the things that we encounter in public health labs are dangerous pathogens. How do we keep them safe from people who use them for harm? That's a real, a lot of countries don't have any concept of keeping things safe from people who want to use them for harm. So we were working on that. And we had several conferences, meetings, what have you, where we would talk about this. And one of the things that people would, my program was involved with looking at the repositories for smallpox viruses. Smallpox is the only virus that we've eradicated so far. We're working on COVID. Smallpox is kept in how many places, do you think, in the world? Two. Where are they?
Switzerland?
Two. That is correct. Where is it?
Switzerland?
No. CDC in Atlanta. That's right, building 24.
Yep.
The CDC in Atlanta. Novosibirsk in Russia. These are the only two known repositories of smallpox. And my program at WHO was required every 2 years to go out and investigate, not investigate, but to inspect, if you will, the smallpox repositories. Are they safe? Are they secure? Could, terrorism happen? Could an accident happen where these viruses could be released inadvertently, not with intent? And so you have to have a lot of gates, guards, guns, et cetera, et cetera. I'm telling you all this because of this polio virus. We were in one meeting, and every time we'd have a meeting on smallpox in WHO, we in WHO, wearing that hat, we're advocating to get rid of all smallpox viruses around the world. How many of you have been vaccinated against smallpox? You have? You were in the military then? No.
I wasn't.
If you were not in the military, okay, chicken pox maybe. Okay. If you're in the military, you probably got smallpox vaccination.
Yep.
Military? If you were in school, grade school, in the 50s, maybe even into the 60s, you got smallpox vaccination. In the 70s, they stopped smallpox vaccination because about 1 in a million of vaccinees would have an adverse effect and they would die.
Wow.
People said, hey, we don't have the disease, nobody's dying from the disease, but we're killing about 1 out of a million with the vaccine, and we really need to do this. So they stopped. So what that means is, I was vaccinated 2005 was the last time, so I'm probably okay. How many years has it been since you got smallpox vaccination?
I got me in 84.
84. So, coming up to 30 years. You probably have partial immunity, but it's, not as strong as it was when you were first vaccinated, but you probably survived. You'd survive if all of us were exposed to the smallpox virus. The rest of you, about 90% of you would die. Okay? 10% would survive, but you certainly have pox all over your face and your body and your hands, and et cetera, the palms of the hand. This is a very, very easily spread virus, and it's very transmissible. It is very durable in the environment, and it's a perfect bioweapon. If you're immunologically naive, and that means you have not seen this virus or anything like it in the past, mortality rate is very, very high. So we would say, get rid of all the smallpox virus in the world. Why do we need it? Of course, the Russians and the Americans were there, and they said, well, we need it for research. We need it for this or that or whatever. You know? If there's another virus that's similar to it, who knows? We may need to go back to the genome. Well, interestingly, they have published the entire genetic code, if you will, for smallpox virus. Theoretically, they can make it in the laboratory. They have made fully active paralytic poliovirus in the lab. You say, whoa, whoa. Doesn't that mean they created life in the lab? Why doesn't that mean they made life in the lab?
Virus aren't alive.
Virus is not alive. It's not alive. You can take the poliovirus, and they have. You can break it down into all the nucleotides and everything else, and you can make it in the laboratory. And it is a real, honest-to-goodness, paralyzed mice, kill them, etc. Poliovirus, it was made out of nothing more than chemicals. Theoretically, could you do the same thing with the smallpox virus? You could. I want you to look at this. Smallpox is huge. It has a much larger genome. It is much more difficult to make. And to synthesize that in a laboratory would be pretty, it's possible, but you'd have to be one of the most sophisticated laboratories in the world. It certainly isn't going to happen in Iran, okay? It isn't going to happen in a developing country or a country that is marginally capable of doing these things. I'm not even sure it could happen in the United States without many years of effort. They have made this virus. So viruses, they're small. They are significant. They are significant to us today, and they're not going to go away. Recently, we had SARS virus. Did you remember that?
Yeah.
Remember that? SARS. And I was at WHO. I was in Geneva. Actually, I was in France at that time, in the WHO office in 2005 when we had SARS in Asia. SARS virus is a respiratory virus. It looks very much like influenza. And we thought it was influenza. And we, WHO, sent a physician to China to work with them because it was in the Guangdong province that they started having these people that died of a disease that looked very much like influenza, but it wasn't influenza. And the Chinese were, it was in the newspapers and it was on the internet. It's interesting. Countries can't hide. And on the internet, it's just kind of brought back the curtain. If you're having something going on in your population, a lot of countries would not tell WHO about it. They'd want to keep trade or travel or something like that. Well, you start to hear these reports. Well, the Chinese weren't telling us what was going on with these patients, but they said that there had been several deaths. They were in an influenza ward. That's what they thought it was. We sent our physician, an Italian guy. He looked and he said, "This is not influenza. We don't know what it is." And 10 days later, he died. The problem with SARS is about 30% of the people that died were where you're going. They were the nurses. They were the doctors. They were the caretakers. So if you're a doc in the box, if you're a nurse in an ER, if you're a nurse practitioner and you're seeing somebody and they've got SARS, guess what?
You called that?
You've just been exposed. Pretty scary stuff. Pretty scary stuff. I mention this just to say that viruses haven't gone away. The significance to public health, to human health. Now, you've heard about Ebola. Ebola, this hemorrhagic fever virus, continue to have problems in Uganda, Central Africa, several countries that have it. DRC right now is having problems with hemorrhagic fever virus and Ebola. And obviously, every year we have our influenza viruses and things like that. talk a little bit about the capsid morphology. Okay? Remember what that capsid was? Protein coat. Yeah. Protein coat provides protection for the viral nucleic acid. Also is a means of attachment, not the only means, but a means. The capsid is made up of things called capsomeres. Okay? And there's three basic shapes. There's helical, polyhedral, and complex. And this is a helical. It's sort of wound, if you will. It's a tobacco mosaic virus. And if you can see this, it's wound around a pore. I don't know if I have, no, I think on the later one. Anyway, this is long tube, very long virus, and it's sort of coiled up, and it's got a hollow portion down there, and this is helical shape. Okay? Now we have polyhedral, many-sided. Okay, this is a many-sided protein coat that encloses this nucleic acid. And I put down here the icosahedron, which has 20 sides. Believe it or not, that's the most common shape for these polyhedral viruses. Okay? Before I leave this, I just want to point out these things. These are the glycoproteins. These things are virally encoded. Okay, this part here, this envelope, comes from the host. These little spiky things sticking out, they are coded for by the virus. They are part of the specificity of that virus for a particular cell. Okay? So these little lollipop things, particularly in influenza viruses, are, I'll show you that later, are very, very important in establishing an infection. So anyway, this is the capsid.
Who would the nuclear capsid be if it were there?
This is the nucleocapsid.
That's the nuclear.
Yeah, it's the protein coat around the nucleic acid. What about the shapes of viruses? Well, these guys are all different. Okay, smallpox virus, very large. The rabies virus looks like a bullet. We've got the bacteriophages that I talked about. These are specific for the E. coli, that particular group, look like the lunar lander. So you can see that the viral shape is hard to, it would be very hard to describe this group in simple terms, whereas other viruses can be. We've said the viral envelope does come from the host cell during viral replication and release. It's composed of a phospholipid bilayer and proteins because that's what the cell envelope is. It's a phospholipid bilayer. Virally encoded glycoproteins or spikes, and they play a role in the host recognition, the attachment specificity. Much of that we've said. Okay, here's another envelope virus. Here are these things that look like lollipops. By the way, you can see this helical. This is like the tobacco mosaic virus. Okay. These things are coded for by the virus. The virus is budding out of the cell, and as it pushes its way through the cell, it encoats itself or enrobes itself with this bilayer, lipid bilayer, and it gets this envelope. Now, interesting, and this is some of the work that I did when I was in my master's. We could see where the influenza virus in the chicken embryo cells, we could see where this virus was going to bud out. You want to know why? These would appear on that monolayer of cells before the virus came out. In other words, the virus was underneath. It encoded for these proteins. They appeared, and before the virus actually budded out of the cell, we knew where it was, because we could see these areas on the surface of the chick embryo fibroblast and say, whoa, that's where those viruses are going to come out. And indeed, in doing electron micrographic studies, we showed that that does happen. I was working with influenza C viruses, and that we could predict where those viruses were going to come out of the cell. Pretty fascinating. Again, the whole question, are viruses really, really primitive? Or are they really, really advanced? And we're talking about some pretty sophisticated things that are going on. Very sophisticated.
It's got the